So I want to start off by going over yesterday's mind stretcher, November 11th. And your first question was, which group contain only molecules that are assembled from smaller organic compounds? So to answer this question, you had to realize that they were talking about polymers when they were talking about the molecules here, and the smaller organic compounds would be monomers. So as you look at your choices, you need to keep that in mind. And polymers we've been talking about are biomolecules. Okay, so if we look at the first one, well, protein definitely is a polymer, a macromolecule. It's a big molecule, and it's made up of monomers. So we know that's a good one. Now, if we think of water, though, it's H2O. It is not a polymer, so we can cross that off right away. So we know that A is not our answer. Okay, proteins, yep, that's good to go. Starches are carbohydrates. So we know starches are macromolecules, are polymers, and they are made up of monomers. So that one's good. Now, carbon dioxide, CO2, that is not a macromolecule. And in number one, we already crossed off water. Okay, so B can't possibly be your answer. So proteins, and both these have proteins, so they're both good to go. We've already crossed off carbon dioxide, so we can cross it off there too. So the only one that doesn't have anything crossed off is C. So let's look at it. Proteins definitely are macromolecules and have monomers. DNA is another word for nucleic acid. We know that's a macromolecule. Fats is another word for lipids, and we know lipids have monomers, and they are macromolecules. And then finally, starch, which are our carbohydrates. So yesterday, you should have picked C. All four of these are the biomolecules we have been studying, and all biomolecules are also macromolecules, which are made up of monomers. And mono means one, poly means many, macro means big, so it all fits together. Okay, the second question you had was, it gives you a list of structures. And this goes back, you did this in 7th grade, where you had to do like a hierarchy and put things in order from either a most complex to simplest or simplest to most complex. So as we look through our list, we have organs, we have cells, we have an organism, organelle, and tissue. So of these lists, there's one thing that's inside of another. So the organelle is the simplest. It can't function on its own. And organelles are part of cells. Nucleus, cytoplasm, Golgi bodies, all the stuff that's inside the cell, organelles. So if we put a bunch of organelles together, what do we get? We get a cell. So the cell's next. That would be the second thing on your list. And if you put a bunch of cells together, what do you get? You get tissue. For example, if I had a bunch of muscle cells put together, I get a muscle. Okay, muscle is a tissue in your body. If I put a bunch of tissue together, what do I get? I get organs. And if I put a bunch of organs together, I get organ systems. And a bunch of organ systems together gives me the most complex on this list, which is the organism. Okay, so those are yesterday, November 11th, mind stretchers explained. So let's dive into today's teach piece. So yesterday we talked about enzymes. Meet our trusty enzyme friends, our enzyme and our substrate. And we talked about reactants and products and how enzymes work. And so today we're going to look at the graph of the activation energy and just graphs in general when it comes to enzymes. So once again, to kind of repeat, a key point is enzymes work best in specific conditions. Okay, they're kind of picky. They're kind of the prima donnas of the body. So if we look any graph, you want to look at the axes first before you start interpreting it. So on this graph, we have temperature. And as we look, we see the temperatures increasing as we move to the right. And then to look at the y-axis, we have rate of reaction. So we're talking about the speed of the reaction, the speed at which our two, I'm missing one of my reactants, the two reactants come together. So at 0 degrees, we can interpret this. There's no reaction happening, okay? Because the rate is at 0, the temperature is at 0, and really, who really wants to do anything at 0 degrees? Okay, that's the time you cuddle up and you stop moving. And molecules agree, and they do the same thing. Okay, so as the temperature increases, what do we see? We see the speed or the rate of the reaction also increases as it rises. Okay, so 30 degrees, it keeps going up, and you have a pretty sharp, every 5 degrees, it goes, and then all of a sudden, boop, and then you reach the top of the peak on your graph, okay? This is like nirvana. This is heaven for the enzyme. This is where the enzyme goes, okay, this is where I'm working. This is my place. And this is where the enzymes can be the most productive, is at the peak of the graph, right here. And for this enzyme, it's 37 degrees. Well, isn't that curious? Because humans, their body temperature is really close to 37 degrees, which kind of makes sense. The enzymes are in our body. We know they're picky, and so it makes sense for them to be adapted to work best in our environment, inside our body, that this would be the temperature range they would work best in. So we have a slow increase of reactions, and then the peak, the most reactions occur right here. But so logic would say, well, if better temperature increases reactions, more temperature will make it even stronger. Not for an enzyme, it doesn't. So what happens is when the temperature increases from this perfect point, you see a drastic drop off. And what happens is enzymes, we call it being denatured. So remember, enzymes have a very specific shape. And when you denature it, it's kind of bent. You change that shape. And if you change the shape, you change the effectiveness of your enzyme. So too high of a temperature, you get a denaturing of the enzyme. And here you have an increased reaction. Okay, so specific. This is just a general overview. So let's dig in and pull out some examples. So here we have two enzymes. We have enzyme Shaz-Adams, and we have enzyme Glencappy. So the blue line is one, whoops, let's back up. Let's go to our axes. We still have temperature, and our temperature is increasing as we go to the right. And we still have rates of reaction on our Y. So as this blue line increases, that's telling me my rate of reaction is increasing to right about here for this enzyme. So this enzyme, and let's roughly say it's probably about 45 degrees. So this enzyme works best at 45 degrees. And then after 45 degrees, it becomes denatured and it stops working. Okay? But if we look at the second enzyme at 0, and notice at 0 degrees, This enzyme there's some reaction going on. But on this enzyme with the purple line, there is nothing happening at 0. So it takes a warmer environment, because you can see as this line increases the rate of reaction until it peaks right here. And remember, the peak is the best for that enzyme. So this enzyme works best at 72 degrees. And then it drops off and is denatured. And denatured, its shape changes. And when you change the shape of an enzyme, you change its effectiveness. So here you have 2 enzymes with different temperature rates. So if I took this enzyme and put it over here, it would stop working. Okay? So it's not only temperature. Yesterday, I also mentioned pH when I was talking about the enzyme pepsin in your stomach versus the enzyme trypsin in your small intestines. Okay, and here's what that looks graphically. So pH, the lower numbers below 7 are the acidic range. 7 is neutral, that's pure water. And then above 7, your higher numbers are basic. So here we're looking at 2 enzymes. So this enzyme A works best in a more acidic environment. And then enzyme B works best in a more basic environment. And by working best, where am I looking? I'm looking at the peak. Okay? That's the optimum point for an enzyme. So it doesn't matter what graph you're looking at. You want to look at the peak of the graph for its best. And if we notice, they also put in temperature down here. So enzyme B also works better at a higher temperature, just above normal. And then enzyme A works just below a normal human body temperature. So enzymes work best in a specific pH. And most of the questions you get will either be about temperature or pH. Okay, so you may be asked to make a graph, and hint, hint, you will be. So you could get a table like this, and we have temperature. And so my x-axis is already set up with temperature, with increasing temperatures as we move to the right. And then my y-axis is the rate of enzyme activity, which is the speed of these collisions that are going on. And the enzyme grabbing them and saying, hey dudes, reactants, let's get our acts together. Let's make some products. Okay, so it's kind of like Battleship 00. Oh, let me get where I can select. So I'm going to take 00, put it on the axis. At 5 degrees, we have a rate of 3. At 10 degrees, it increases to 10, and this probably should be lower. My circles are kind of chunky monkeys, though. 15 degrees, we have our rate of reaction goes to 25. At 20 degrees, our reaction rate jumps to 40. 25 degrees, it jumps to 65. That's about 65. At 30 degrees, it jumps to 85. And at 35 degrees, we have 100 reaction, 100% is happening. And then at 40 degrees, look what happens. Bam! All the way down to here. And then 45, nothing's happening again. Okay, so here's the increasing reaction rate. And so the optimum for ATP synthase would be at 35 degrees. Now, if you think about it, ATP is energy the cells make when you break down the food. ATP is released as energy. And think about it. So here, this is when you have a fever. If we're talking about the human body. So this is fever range. And if your enzyme that delivers energy is denatured and doesn't combine for the reaction, so your body has energy, does it make sense now when you have a fever that you're tired, you're run down, you have no energy, and it's all due to an enzyme? Okay, so that's today's lesson on enzymes and reading their graphs.